Mercosur. I'm Daniel Treffer, founder and CEO of METT. First of all, I would like to thank the organizer Merck for inviting me to present at their workshop. I will talk about my invention, vacuum compression molding, today. I invented the process to prepare samples for investigations on polymer melts, and it turned out to be of great use for pharmaceutical formulation development. It is beneficial to find ways to overcome the often quoted solubility challenge, or rapidly prototype drug-eluting implants. Let's start with the solubility perspective. The pharmacopoeia classifies the solubility from freely soluble to practically insoluble with intermediate levels. Commonly known examples for freely soluble substances are salts or sugar with solubilities in the gram per milliliter range. We dissolve them every day, that's no big deal. Since we are talking about the challenge, we are interested in the other side, in practically insoluble substances. Famous buildings like the Parthenon in Greece are built to last for eternity. Their material of construction is marble, which is practically insoluble according to the classification. Marble has a solubility of 10 micrograms per milliliter. Here comes the problem. Insoluble APIs are far less soluble than marble. Itraconazole, for example, has 10,000 times lower solubility than marble. What does that mean? Should we be worried when it starts to rain in Greece? Let's do some math to find out. The Parthenon itself consists of 20,000 tons of marble. What do you think? How much water would be required to dissolve the Parthenon? It's a vast number, so I tried to correlate it to something big. I thought Olympic pools would fit the example, make it more visible. We would need 800,000 Olympic pools to dissolve the Parthenon. I believe we don't have to worry about rain in Greece, as there are also other mass transfer parameters to consider. Marble is by far the best choice to build something for eternity. We are not here to destroy famous antique buildings anyway. We want to talk about pharmaceuticals. Let's look at numbers of marketed APIs. Itraconazole has a solubility of 1 nanogram per milliliter. For a typical dose of 100 milligrams of itraconazole, we would need roughly 100 liters of water to dissolve one dose. Another example is fenofibrate. Here we would even need 205 liters to dissolve one dose. Some of you might already see the problem here. We cannot have enough fluid in our bodies to dissolve one dose. So what's the trick? How can we dissolve enough API in our GI tract to make it accessible for the body? As with every trick, one has to understand the basics before we can unfold the magic. Very often, it's the lattice energy of a crystal that holds the molecules together and prevents dissolution. By processing the crystals with an amorphous carrier via hot melt extrusion, we can produce solid dispersions with reduced lattice energy and increased solubility. Different types are possible. So crystalline API clusters within an amorphous carrier, or molecules closely arranged in amorphous clusters, which are embedded inside the polymer, or most preferred option for solubility enhancement is a glassy solution. In this case, single molecules well distributed in the polymer, there is simply no lattice energy to overcome. The API release will follow the release behavior of the polymer. When the polymer dissolves, a single molecule, your single API molecule, will just drop into the dissolution media and will be available for the body. Why is this so important? I assume this drug development process map is something that you're all familiar with, starting from early drug discovery, going down via several clinical stages to bring products onto the market. Most of the APIs in the development are classified as poorly soluble APIs. I want to highlight the compounds row in this table. One has to screen many different APIs which are available in small quantities those are very often new chemical entities, to find suitable and safe candidates to treat a certain disease. You're most likely familiar with the typical screening methods that have been used so far, with all their benefits and also with their challenges. We need an extrusion-like method in the early stages to allow easy scale-up to manufacturing down the road. This was only possible by the use of lossy miniaturized extrusion equipment in the past. Many times this route was not pursued as simply not enough material was available to do that. My invention now extends the playgrounds for the scientists in the field. VCM fulfills all the listed requirements. With vacuum compression molding, one can prepare samples starting from a few milligrams, comparable to solvent casting, and one can go up to the low gram range, always mimicking hot melt extrusion behavior. One obtains perfectly precise shaped samples in a lossless way, perfectly suited for the development tasks. Let me show you how it works. This here is the VCM tool. The vacuum compression molding process transforms any thermoplastic powder into a solid specimen with defined dimensions. Parallel surfaces, no air or moisture inclusions, and the best of it, it works lossless. Put 1 gram of material in on one side, and after processing, you will get 1 gram of material out. There are no losses. Let us look at the process map again. How does it look when we use the VCM technology? We can help to reduce the costs and time efforts in the very early stages. And we hear from our customers that they have a higher success rate in bringing their developments through the early stages, as they can gain deeper insights into their formulations right in the beginning. Using larger VCM dimensions with samples up to 5 grams, it's also possible to bridge towards pilot scale investigations, providing enough materials for tablet compaction, for example. Let me show you how I came up with my invention. I had a huge problem in the beginning of my PhD. I was not able to deliver the expected results. I was working on hot melt extrusion, my specific task, melt rheology of pharma polymers, and here are some impressions of my samples. Moisture, degradation, shrinkage, and brittle samples did not allow me to achieve reproducible measurements from those samples. And you can clearly tell I was having a hard time back then. I had to develop something new. In February 2014, a colleague of mine gave me this new material, a non-stick PTFE foil reinforced with glass fiber. I had this material in my hand, I rolled it up, and it sparked an idea. I knew it was perfect for lining a sample chamber. You can see the first sketch here, already showing the main innovation, a fully PTFE lined sample chamber adaptable for all processing stages. This sketch is considered to be the birth of melt prep, and it developed into a sophisticated solution, what we call VCM Essentials quickly. Here you can see a photo of the VCM Essentials. It's the platform that I have designed to make the VCM process easy to use. On the left-hand side is the heating unit, the vacuum unit is in the back, and the cooling unit is on the right-hand side. The VCM tools are manually placed on the heating unit or cooling unit as they go through the processing steps. Let me show you in an illustrative video how it works. This is the heart of the VCM technology. A PTFE foil is rolled up to cover the lateral surface of the VCM chamber. A disc-shaped foil covers the bottom. It is lightweight and can be filled on an analytical scale. After filling, the opening is covered with another disc-shaped foil on top. The foil arrangement allows the piston to go up and down. The volume of the chamber is adaptable and allows the chamber to follow the material behavior during processing. The VCM tool is then connected to the vacuum unit. The air is drawn out from the chamber and the powder is compacted. Subsequently, the VCM tool is placed on the hot plate to initiate the melting. The powdery material forms a homogeneous melt. As a rule of thumb, this takes about 5 minutes. Once completed, the VCM tool is transferred to the cooling unit to cool it down to ambient conditions. Once cold, it is taken apart and the sample can be released with ease. The BTFE foils can simply be peeled off like stickers from the sample. Here you can see the entire VCM Essentials again. A typical VCM sample preparation takes less than 10 minutes. Heating is performed in some minutes. It can go up to 300 degrees Celsius, making it suitable for most pharma applications. Cooling generally also only takes some minutes. At the end of the operation, there is no tedious cleaning required, making the process time efficient and lossless. Let's dive deeper into the VCM technology. Maybe you already wondered how the API is mixed into the carrier material. Unlike an extruder, there is no mechanical mixing going on. In other words, there are no shear forces, no significant shear forces. With vacuum compression molding, you only compress and melt the material. To illustrate the mixing mechanisms, we prepared an example. If one puts multicolored pellets into the sample chamber and runs the VCM process, one will obtain a disc without any air inclusions. But you can clearly see the phase boundaries between the individual pellets. There are no shear forces and no macroscopic mixing. How can we obtain a homogeneous result then? By using powders instead of pellets to enable mixing via diffusion. We used an orange color dye on HBM-CS powder as a powder blend to illustrate that. As a result, a good fraction of the color tracer diffused already inside the polymer disc, but there are still some agglomerates visible. That's why homogeneous powder mixtures as a starting material are so important for our process. To improve the homogeneity, we have to empower diffusion by reducing the length scale further. The way how we usually recommend to do so is milling, preferably cryomilling. Reducing the particle size before processing via VCM will let you obtain an extrusion-like result, an amorphous transparent disc. Another option to milling is solvent casting. We dissolve the mixture in a suitable solvent and let the solution evaporate. Took the remaining film off the petri dish and put it into a VCM tool. It will also shape into a completely homogeneous disc, which is extrusion-like and suitable for further investigations. One thing I'm very proud of is the small-scale development that we did. I kicked the VCM tool off with 25mm disc diameter in 2014. My former colleagues at the RCPE were requesting smaller dimensions for their investigations. So we reduced it down to 5mm to make it suitable for DSC and later even to 2mm samples. You can use those small dimensions for the screenings of implants, for example. 1 to 50 milligrams are sufficient to prepare a prototype of such a subcutaneous implant. But we did not stop there. We recognized the need for even smaller tools with even lower material requirements. Let's imagine samples with smaller diameters and a length to diameter ratio of 1 millimeter. We used 1 gram per cubic centimeter as a density, which is an educated guess for a polymer density. So when we look now on a 2 millimeter sample, we would only need 6 milligrams to produce a 2x2 sample. For a 1 millimeter, we only need 3 milligrams. For 0.8 millimeter diameter, it's already less than a milligram. If this is still too much material for you, or if you're interested on designing an intraocular implant, one can use our smallest dimensions in the portfolio, which would be 0.5 or 0.3 millimeter. And then you reach down into the microgram range. The entire portfolio of dimensions you can produce with our VCM tools is shown on this slide. You can make disc-shaped samples with a diameter between 25 mm and 0.3 mm. We also offer a VCM bar tool that allows the production of a rectangular bar with 10 by 40 mm. This is used for characterization of mechanical properties a lot. Do you like this flawless samples here on this rotating video? Each of them was produced in less than 10 minutes. Let me show you some of the main applications. I already mentioned it all started with sample preparation for rheology. But my colleagues were interested in other analytics, DMA, solubility testing, dissolution testing, and much more. It soon turned out that our customers are much more creative in using our tools than we are. Let me show you some examples how scientists gained advantage using our VCM technology. I want to start with one of the most frequent questions that I receive. Are VCM samples comparable to hot melt extrusion? At the AAPS in 2019, Thomas Kipping from Merck Germany, also known as Millipore Sigma in the US, demonstrated exactly. They compared analytical results from VCM samples to hot melt extruded samples. I would like to highlight one part of their studies, the BXRD scans. If you compare the left graph with the right graph, you barely see a difference. That means VCM is also yielding an extrusion-like result in terms of BXRD data. This was already indicating that it's of great help for formulation screening. Based on this initial data, a comprehensive study was conducted by a PhD student of Professor Michael Radka. In this study, the model compound Indomethacin was used to investigate if hot melt extrusion and VCM yield similar results. On this slide are images of VCM samples with 8 mm diameter with some common carriers, including Bartek MXB. The samples went through intensive characterization, showing comparable data. The paper is open access and is linked on the QR code on the slide. I would like to highlight the dissolution data. The VCM samples were milled and then subjected to dissolution. One can see significant differences between the carriers. One unique feature that I would like to point out here is the performance of Bartek MXB, because you can see a very high supersaturation, which is obtained over a prolonged period of time without recrystallization of the drug. The same formulations were also processed via hot melt extrusion, milled and then subjected to dissolution. What you can see here is a very good agreement between VCM as a screening approach, requiring only 1 gram of material per formulation, and the processing approach, hot melt extrusion. This was also confirmed with the similarity factors. The dissolution data before was generated on milled material, but there is no need to mill them. You can work with the VCM samples directly. The nice thing is you get a defined surface area. You can think about this approach similar to intrinsic dissolution, because you always start off with the same surface area. To illustrate that, we have made an example with 2 different carriers with the same amount of phenofibrate. So we used Soluble and the PVP with 10% loading. You can immediately see in this time lapse here that the phenofibrate tablet on the right side is dissolving super quickly, whereas on the other side the tablet is still intact, just lying at the bottom of the dissolution vessel and dissolving very slowly, swelling a bit and then eroding. This is also the explanation for the difference in the dissolution profile what we see. With Soluble, you have the zero order release. With BVP, you have the first order release, the fast one. And this is a very nice way to test the performance of your formulation. You can use this information and then tailor the surface area of your dosage form to your requirements. Along the same lines, there was a publication released just before Christmas 2020 about microscopic erosion time testing from EPWI in Germany. It uses a microscope and a VCM sample that is embedded together within dissolution media between two cover glasses. Unlike the dissolution tester, they forced a two-dimensional stationary dissolution that allowed them to track the erosion under a microscope. By preparing multiple samples with various API content, their setup reveals the dispersibility limit of a carrier API combination with minimum effort. Let's move on to the next example. Are you interested in working on multi-layer systems? VCM also allows you to fuse multiple layers to one specimen. So you can prepare various layers in advance one by one and load them into the VCM chamber and process them in another cycle into one single specimen. You obtain a multi-layer sample with distinct, sharp interfaces. One application for multi-layer systems are similar systems to the contraceptive intravaginal ring that you may have heard of, the NuVa ring. A group in Graz wanted to study such a structure and try to mimic the complex structure of the NuVa ring. The NuVa ring uses a drug-loaded core with a surrounded diffusion barrier. So you have a core and the surrounding diffusion barrier, and this limits the drug release. The final VCM sample that they ended up with was a sandwich of two 100 micrometer thin diffusion barriers filled with a drug-loaded core. Then they covered the lateral surface with barrier glue, used optical coherence tomography to check for the interfaces and see if the diffusion barriers are uniform in thickness. And afterwards, they subjected the samples and a co-extruded version to dissolution. So what you see here, this is giving a very good match. But what does that mean? It's empowering the single researcher. One single scientist can perform the VCM process, making the samples the first half of the day and has still the afternoon to focus on research. Whereas with co-extrusion, one has to conduct quite some process development. We're talking about weeks and kilograms of material before a good prototype of a new formulation can be obtained. So this is a huge advantage. Rheology on VCM samples can help to find the processing window of your extruder. During my PhD, I realized quickly it can only help if you receive reproducible data. During viscosity measurement, shear is applied to a molten sample and the required force is recorded. A rheometer assumes a homogeneous sample. Inhomogeneities, like bubbles, lead to falsified data. Compare these two images, taken just before the measurement. The perfect sample on the left, and on the right a sample with many bubbles, without proper sample preparation. How can it be applied for hot melt extrusion development? In general, the viscosity of an API-loaded system is decreasing with increasing API content. This is also known as the plasticizing effect. Adding 30% of API reduces the viscosity of 3000 Pascal seconds at 170 degrees C to only 5 Pascal seconds. The time-temperature superposition allows you to shift those curves to different temperature levels. You can predict operating temperature before starting the extruder. In this case, it predicted a temperature decrease from 170 degrees to 100 degrees C. And the best of it, it worked just perfectly when you turned on the extruder, when we turned on the extruder. This is very powerful. Less material is wasted during process development, and you gain valuable know-how to reduce the operating temperature to avoid API degradation. People usually ask me how much material is required. Will it save us material? We have depicted the requirements here. Standard rheology tests are done 3 times for each API concentration. 12 samples weigh around 12 grams with 1.8 grams of API. To reduce the amount of material, you can do micro-rheology. 12 samples with 8 mm diameter require 300 mg of material and only 45 mg of API. Imagine operating an extruder at the lowest throughput that you can find, let's say 20 grams per hour. You have less than a minute to find your operating parameters before you wasted the amount you would need for rheology. If this is still too much, even smaller sizes are available, but they also require experienced rheologists to perform the measurement. The material requirement can be as little as 10 milligram, corresponding to extrusion times of a few seconds. Is it worth spending a bit of effort in rheology? I think the possible savings, even with standard rheology, are enormous. Rheology is certainly something that I can recommend for process development. Let's move on. This study was conducted by the group of Claire Strahan in Finland. This example is research to reduce the risk of surface nucleation of pure APIs. The starting point is a 20 mm VCM disk out of 100% API, in this case Indomethacin, with a height of 1.7 mm. Air humidity is causing stability issues, and the VCM samples immediately show surface recrystallization. But the smooth surface of the VCM samples allowed them to put a protective coating on top. It's just 1 micrometer thick. And you can see here a comparison on this slide. On top, you can see the uncoated samples, and on the bottom, the coated ones. What you can tell from the images, the surface of the uncoated starts to recrystallize on the very first day, while the coating increases the API stability significantly. Let's move on to the last application that I wanted to show today. It's about VCM in combination with spectroscopy. We can mount 2 processing sensors into our dedicated VCM spectroscopy chamber. This is used for studying PAT approaches offline. You don't need to have an extruder. You just mount your process sensors and fill the chamber with the desired material. It easily allows you to develop your chemometric model or choose the suitable spectroscopic method for your formulation. This also requires just a couple of grams per run, significantly lowering the material requirements. You have seen many different applications with the easy-to-use VCM tool. I mentioned earlier, our customers are always more creative in using our tools than we are. So I'm excited what you are going to do next. Finally, I'll give you a short overview of the entire METT portfolio. You already know the VCM technology for the lossless sample preparation, but there is more than that. The second innovation is called the V-chamber. The V-chamber turns a regular hot plate into a vacuum oven. Place your sample on the hot plate, then put the V-chamber over it, and you can apply vacuum and melt or dry your sample directly under vacuum on your hot plate. You can also think of melting it where you measure it, for example, on the rheometer ground plate. And last but not least, Extrusion, a useful tool for scale-up and troubleshooting of continuous processes. It's a camera-based system allowing you investigations of residence time distributions, giving you insights into mixing and degradation. It can also help to trace down content uniformity problems. How does it work? You apply a color tracer at the inlet and point the camera to the outlet. The supplied software uses the color response to to provide the analysis for you. You get the residence time distribution evaluated by a click of a button. Then you can compare and trace down problems that you may have seen during the processing. We have seen this slide earlier. I would like to show here where Extrusion comes into play. It will help you to go through different processing scales, from lab scale to pilot scale and finally to production scale with ease and confidence. That's about the portfolio. I would be delighted to talk about your challenges and explore ways of overcoming them together. That's why I would like to invite you to book a free video demo where I can share some more of my insights and prepare a sample with your material live. You can conveniently book via our webpage, meltprep.com/contact, or contact me via my contact details shown on this slide. I would like to thank my team for their help during the preparation of this presentation and the recording. Thank you very much for your interest. Arigato.